Thank you very much. So that will be the last talk, and it is not a speciation talk, I should say. I'm really talking about the question of Maid Choice preference, and of course I prepared a slide saying why that should be important, but I think since we are so much advanced in the conference, there's no need that I have to defend this. So basically, the house mouse is not, okay, so we come to the end to this. So Maid Choice is really what I want to deal with today. So to remind you that Maid Choice is of course an important component of speciation models, and this is, I just picked out two of them, and this is the one by Ulf and Michael, where they basically in one type of simulation they assume that the Maid Choice is connected to the ecological character, and Ulf actually explained this model already, and you can get branching under these conditions. But importantly, they showed also that it is possible to, under certain conditions, you find that the character evolves independently of the ecological character and serves as Maid Choice. So that's one way of looking at Maid Choice in the speciation model. Another one is, of course, using the Maid Choice or the signal, so to speak, as a signal for the quality of an animal in a certain habitat, and that's a model by Van Doorn, which was published this year, and again he finds speciation working well under these conditions. So that's just to remind you there's a whole spectrum of how to look at Maid Choice under speciation models. Since Ulf didn't show our book, I have to show So in this book, which was actually the outcome of a workshop on speciation, which we had 8 or 9 or 10 years ago, I don't remember, actually here in Luxembourg. And in this book we had, in the epilogue, we had a box on future empirical research, and one of them was to say, okay, a further alternative, so instead of evolution of Maid Choice, a further alternative is that an assortative mating system is already present in every system, and so there are many reasons for that. And genetic assortativeness can be considered as something that is already built into the system of sexually reproducing species, and that it does not have to evolve newly during a speciation process. Now, of course, that sort of asks for experiments to test this. Okay, so we use the house mouse as a model system for evolutionary studies, and just to introduce this quickly, it's actually a beautiful model, and it's sad that relatively few people still work on it. So the house mouse, so there's a very detailed phylogeography available, so we know it comes somewhere from the Indian subcontinent, has spread in various cycles across the Earth. So the first one was into Asia and Western Europe, and that is the recognized subspecies domesticus and musculus. And the domesticus population then spread further across the Atlantic and into Africa, in fact, and also into Australia. So we have different time horizons, and in fact we have fossils in some cases where we can actually date the times where they arrived at certain areas. And that's particularly true for the populations that we use frequently, and which is also in this study. So we use a population from France, from Massif Central, and one from the Cologne-Bonn area. And the history of these populations is that currently it seems that the mouse arrived about 1,000 years BC with the first Phoenician trades across the Mediterranean, and then from there spread very quickly across Western Europe. And so we can have a sort of time stamp on it and say, okay, the estimated divergence time between these populations is a maximum of 3,000 years. It could be less, but we take the maximum, assuming that the spread here has been indeed relatively quick, and then little subsequent gene flow. In fact, we can show that there is a good differentiation between these populations. It's also an old study. So we used here, this is a microsatellite allele sharing tree, which is very sensitive to show population differentiation. And here you see, for example, the French and the German population. So each is an individual here. Basically all are clustered to their origin. So there is, as far as we can say, very little ongoing gene flow between them. So they are, let's treat them as separated populations which are split 3,000 years ago. Now our approach to this, so we use these sort of systems for approaching various questions, and I want to go into one of them just to show you how well we understand the system already. So we use a broad attack on this, so to speak. We look, our main interest is to understand the genetics of adaptation in the house mouse. And that comes a little bit back to this question which was raised, I don't know by whom, whether adaptation and speciation is the same or different. I think it's the same, and well, it has something to do with it. So what we do is we use one phenotype-independent approach. We look for selective sweeps, simply based on randomly selected polymorphic markers. So we have no preconceived ideas which locus we're looking. But then we look also excessively at changes of the gene expression. And we have also phenotype-related approaches where we have actually phenotype differences and then map the genes between them. So coming quickly to these selective sweeps, I think, again, in this audience, I don't have to explain this in much detail. The whole idea of a selective sweep is that in a polymorphic population, if you have a beneficial mutation, that is fixed in the population, and of course the flanking markers, which can be neutral markers, are also fixed, and that allows us to basically to screen systematically for such regions. And of course the window of a selective sweep depends on recombination rate, selection coefficient, mutation rate, and so on. So for example, we did one of the screens we did was with 960 microsatellite loci across the genome, and we find actually quite a few candidate loci among them, and then you have to do lots of additional tests and be sure that this is not an artifact of whatever. There are many artifact possibilities. So out of 30 initial candidate loci, we have 5 of them we really trust that they are true selective sweeps. So I think 3 in France and 2 in Germany. And in fact, because that's so systematic and because we have a timestamp on these populations, we can actually estimate average frequencies of positive selection events. So our screen has covered about 1% of the mouse genome, and then we can calculate that, well, we found in one lineage, we found 2, in the other lineage, we found 3 significant sweeps. So it's about 200 to 300 sweeps have occurred in 3,000 years, which in mouse terms is about 9,000 generations. So we end up with an approximate average sweep frequency of 1 per 30 to 45 generations. And also I should say the average window size is lower than 100 kb, so it's normally located within a single gene. We should say, because we are very stringent with the statistics, this is clearly an underestimate. So I don't know how many of you know the discussion about the maximum allowable positive selection in a population. Depending on who is modeling this, this is high or low. But this, I should say, I think at least, is the first real measurement in a natural population. Estimates have also been derived from a species comparison, which also arrive at these measures. Okay, that was basically the background to our populations. So what I wanted to say is there are separated populations. They have undergone separate routes of adaptation to their environments, although their environments are actually not tremendously different. So Massif Central is, of course, an elevated area, so it's not necessarily much warmer than the Cologne-Bonn area. Okay, so now the assortative mating experiment. What we have done is we use approximately 10 pairs from the French and the German population. We put them together in a room and allow free mating. And then run the experiment for 2 to 3 generations and then do paternity tests on all animals. And then, of course, one can evaluate the mate choice on the basis of the offspring produced. So this is probably the most direct measurement of success of reproduction. So that's a little bit complex. So we have the experimental setup. So we have run 2 such experiments, and each of them was run in 2 separate rooms. So the first one, well, these are details here. What I want to point out, okay, there's an initial density of mice. It's the same in both rooms, and then because they run for different times, they have a different end densities and so on. So importantly, I think the first experiment, I show the room first and then you see what I mean. Hope it's reasonably visible. So this is the room. It's about 20 square meters. Then we have little borders here so that they can establish territories, but they of course can run around them. These is places where they can build nests. And in the first experiment, we used this wall separating the two halves of the rooms, so that we put the German on one side, the French on the other side, and then they had about 1 week. Okay. They had 1 week of getting sort of used to the new situation. Then we opened the wall and let them interact freely. I should say the first, so then we of course recorded very carefully when the first litters were born, and there was no successful mating during the time where they were separated. All matings occurred afterwards. And then they actually mixed throughout the room. Okay, there are a few other observations that we made. There is a frequent occurrence of multiple paternity. I think this is across all experiments, and while it's both the same for the German and the French animals, there's about around 30% multiple paternities. So when we then count paternities, each of them is counted independently. 30% is also what one tends to find under natural conditions. There's a large variance in individual mating success of males, and there's fierce fights among the males, so we know that there is a pressure for the males to get the paternities, basically. Okay, also this is a result across the whole experiment. There are no indications for any hybrid sterility effects. So basically, the offspring number is not significantly different whether they are pure or whether they're hybrid animals in the end. It's just as a background, so there's no Dubchan-Skimula incompatibility as far as we can say. Okay, so the first experiment with 1 week initial separation, then we look at the proportion of F1 offspring, and then, okay, this is room A, room B, and then the colors basically say, okay, this is assortative mating is GG, so it's German, or the FF is French, and then these are the two alternative disassortative mating possibilities. I distinguish here between all animals, and then among those where we have removed those animals which carry the T haplotype. T haplotype is a selfish drive element which is known to influence mating preferences, and it's always present when you use wild-caught mice. What we did in this case, then T haplotype will be present. We didn't want to exclude this because we wanted to have the experiment as natural as possible. But what we can say is that the T haplotype does indeed shift a little bit the effects. But here we see clearly in the first experiment, we see it's pretty well assortative. 43 out of 61 of all animals assortative, or 35 out of 42 T haplotypes removed. So this means the first conclusion, yes, there is differential assortative mating between the allopatric populations. And the underlying mechanism obviously must evolve fast because there are less than 3,000 years of separation. Okay, then second experiment, so there was no initial separation of the two populations. And under these conditions, what we then see is basically there is no assortative mating anymore. So if they don't have the time to sort of get as used to their own population, and they don't have the chance in the cages where we keep them, certainly not the males because they have to be kept individually, otherwise they kill each other. So there is basically no indication of assortative mating. If anything, then there is disassortative mating. There's actually fewer than random. So under this, but the experiment had the advantage in this case, of course, to produce lots of F1 offspring, of hybrid offspring. And that's the reason why we run this experiment longer, because then we wanted to know, okay, what are these hybrid animals are doing in terms of assortative mating. Okay, if we look at this and look at all possible combinations and then ask how often mating was successful, so each of these numbers is a single mating. It can have multiple offspring per mating, of course, but this is single matings. So we see that there is, in fact, a strong assortative mating under these conditions, even among the pure animals, because don't forget, now they had enough time to get accustomed to whatever their own mating signals are, and the Germans among are very assortative. Then here are a few which are disassortative, the 5 here, but here the hybrids, GF, so this means the female had a German mother and a French father. They mate assortatively, FG also, FF also. There are fewer French males because they had too many T haplotypes were brought in by the French. So we have 60 to 5, so it's clearly highly significantly assortative. But now the interesting thing comes when we look further and ask what happens under these conditions where there's one hybrid and one pure. So if you look at the GF female and G male never mate with each other. However, if we look at the GF with the FF male, we have enormous numbers of matings. And we call this paternally matched because they have the same paternal population background. It's not the same father, they have the same population background, the French background. Now, and if you go through this, actually it works very well in both directions. Not only, so females presumably choose, so they choose males which have the same paternal background, but it works also for the males. If you look here, for example, the males are accepted by females when they have the same paternal population background. So paternally matched are 10, and non-matched, they're none. So basically what we can say, can be interpreted as a paternal imprint, and if we do this across all animals, then we have 127 out of 151 is of course highly significant, and T haplotype removed becomes even more significant. 98 out of 106 animals mate according to paternal matching. So that includes, of course, the assortative matings. Okay, conclusion 2. Assortative mating in the F0 generation requires a phase where the animals can familiarize with each other, so there is a learning phase required to do the right choice. And that puts a little bit into question previous attempts to measure mating preferences by not having this learning phase included. We don't know yet how important this is. It's sort of very new results, but it's certainly a possibility. And then, completely unexpected, I would say, there's a strong paternal imprint on mate choice preference. How is it done? I can only speculate. There could be odor signals or pheromones. And of course, we know for the mouse that there is both MHC constitution and major urinary proteins provides recognition cues for mating. However, under our conditions, so both of these represent polymorphic multigene families, which are very unlikely to have diagnostically been replaced between these closely related species. So we know this already now for the MHC. We sequenced quite a few of them. MUPAs have not been done so far, but it's very unlikely that they have a diagnostic difference for them. And most importantly, both are not known to be paternally imprinted, although I heard that the MUPs may be paternally imprinted, but we have to see. So it's one possibility. We cannot, maybe there is something behind this, but I think, so these cues are more likely to signal quality of individuals, males within populations, but possibly not between populations. I prefer the other idea that there's ultrasonic vocalization. So mice can sing, it's known since a long time. They communicate in ultrasound, and they have actually complex song patterns, which were never very carefully recorded. So accordingly, it has not been implicated in differential mate choice so far in mice, but it has also not been, as far as I know, not been specifically tested. Interestingly, it was found earlier this year, it was published that there's a paternal bias of gene expression. There's a paternal imprinting of gene expression in the adult cortex and the hypothalamus of the mice. And these could be involved both in zone recognition and learning. So it's actually well known from birds that they can do that. And so why shouldn't it also be possible in mice? Why? Well, why is there such a fast divergence of mating preferences in allopatry? Sorry, I have no answer. Why is it paternally imprinted? Also no answer. But something for you to think about a beer tonight. And this, with this winter impression from Plön, I should say Inka Montero was a PhD student who did the work. She was supervised by Maike Teschke. And special thanks to Barbara Koenig, who helped us with setting up these common environment experiments. Sorry, I didn't fully.
That showed that in the same farm, we have two teams that have different photos and have so it can happen even in close nature.
Yeah, I don't know the study.
So I have a question, the two populations are the same subspecies?
Oh yeah, they're both domesticus, right?
It would have been interesting to mix the population of the two animals, where you mix the population at the image and let them put the reef, and then find out into the mixture of the two populations. But just to find out whether it's the geographical effect that made the divergence between the two populations, or it's just the fact of being familiarized with potential targets.
Yeah, but we think it's clearly a familiarization question because in the second experiment they were not separate, but they had no chance to familiarize, so they produced a lot of hybrid offspring, but the pure offspring among them still mated then assortatively very strongly. So there was a learning effect, certainly.
I think maybe over there. Yeah.
More comment and question. Paternal imprinting should cause much stronger sexual selection than the phenotype matching, so I'm wondering in allopathy, strong directional selection because of paternal imprinting would allow for rapid.
Yeah, I mean, basically, yes, paternal imprinting should reinforce well, not reinforce, I should be careful with words, but we should make a stronger effect on retaining an assortative mating pair. So it's probably against splitting tendencies, I would say. That
it causes strong sexual selection because you're imprinting on a very skewed segment of the population.
In this case, I would not be sure what sexual selection and assortative mating, what the difference would be in this case, because there's no obvious signals that. Sorry?
You're imprinting on males that have skewed mating success, you're causing directional preference.
Yeah, I call this assortative, not a sexual selection, but maybe.
But I guess the certainty would be causing strong sexual selection.
Yeah, okay.
Thank you. We have further questions.
So regarding the capacity to make French on the males, what do you think would have happened if you put the French females with the German males, or even vice versa, before releasing carriers? Do you think it would imprint this as a tissue difference?
So again, the German.
In your first experiment, you put French females and French males
Together, yeah. Yeah.
What would have happened if you put French females?
Oh, if you You mean if we sort of have the learning phase with the wrong partner? It's a good question. In birds, we know it works, but not very well. So zebra finches, these experiments have been done extensively, and zebra finches prefer to learn their species-specific songs, but can also learn other songs. That's right.
We also I'll show you where the cloning Dawson. I was wondering whether you had sequenced Dawson in a German stock.
No, we have not specifically looked at this, I must say. Darwin is only one of the MUPs, and why should this be particularly important, I don't know.
Because it's Diethard actually.
And, well, it was tested only in short smell tests, so without learning phase. So we don't know. So it's certainly recognized by the females, but we don't know whether it's recognized in the context of mating preferences or defending territories or whatever. So it's open at the moment, I would say.
It is monomorphic, and what it actually does is it turns on the imprinting to the other polymorphic markers. So if they diverge slightly with Dawson, then that might explain why one will work on German stocks and one will work on French stocks.
Yeah, we will certainly follow all this up, that's right.
Do you have any more questions? There are more questions over here.
Can I ask please, the experiment was
Can
made.
you wait for the microphone? It's a bit difficult to get. Could you also stand up?
Could I ask please, it was made on inbred strains or it was wild?
No, these are wild mice, so they're wild caught and then kept in, when we did the experiment, they were kept for 2 generations under outbreeding conditions in the lab. So they're real wild samples here.
All right.
I'm interested in the T locus that's so important in the system. It's the same T locus that Lewington and Dunn studied 50 years ago to talk about group selection.
That's the same locus, but not necessarily the same allele. There are quite a number of different alleles.
So does this one tend to cause the extinction of deans locally? Instability I don't know how to put it, but if it does, then there's an interaction going on.
So it's a selfish drive element, so which when homozygous is lethal, but it tries to drive itself through in the population. So the gametes carrying the T have a much higher probability to go into the next generation. So theoretically, of course, it can drive populations to extinction, but as far as I know, all attempts to try to mimic this under these natural conditions never went to extinction. The T locus, this must be a more complicated situation. Also in our case, it increased in frequency only in one of the rooms, but not in the other rooms. So.
Okay, so